An observational study is a non-interventional study where researchers observe what happens in routine care. Treatment is not assigned through a study protocol. Patients receive the care their clinician would normally choose, and the study captures exposures, outcomes, and relevant clinical information as they occur. The defining feature is not the data source. An observational study may use electronic health records, insurance claims, hospital records, pharmacy data, disease registries, or other real-world data. What defines it is that the investigator is not controlling treatment allocation in the same way as a randomized controlled trial. This has consequences for the whole study. Treatment decisions are made in routine care, so the groups being compared may already differ before the analysis begins. Those differences affect how the study should be designed, how the data should be analyzed, and how cautious the interpretation needs to be. Different real-world data sources bring different limitations. Claims data may give broad coverage of treatment patterns, but often with limited clinical detail. A disease registry may capture richer disease information, but may have tighter inclusion rules and its own missing data issues. Many problems start when the study question is too loose. Exposure, outcome, and baseline period need to be defined before modeling begins. A treatment comparison might identify exposure from prescribing records and outcomes from hospital events. That can sound simple until timing windows do not align, or a dosing change is visible in one system but not another. Observational studies can take several different forms. In a cohort study, groups are followed over time and outcomes are compared between people with different exposures or treatments. In a case control study, the analysis starts with the outcome and looks back at prior exposures. Cross-sectional studies provide a snapshot at one point in time. Case series and registries can help describe disease course, treatment use, or uncommon settings, where the immediate aim is to generate hypotheses. These studies are useful because controlled trials cannot answer every question on their own. Observational studies can examine treatment use in broader patient populations over longer periods and in day-to-day clinical settings where adherence, comorbidities, and treatment switching are part of normal care. They may also be relevant when randomized trials are difficult to run for ethical or practical reasons, including some rare disease settings. The main analytical challenge is bias. Clinicians prescribe based on disease severity, prior response, comorbidities, access, and many other factors. If one treatment is more often given to sicker patients, a crude comparison may make that treatment look worse, even when the treatment itself is not the reason. Measurement can add further uncertainty. Symptoms may be recorded differently between sites. Lab values may come from different systems. Outcomes can be misclassified, and missing data is common. These issues need to be understood before the analysis is treated as evidence. Baseline review is one of the most important steps. Researchers need to examine how groups differ in age, disease severity, prior treatment, comorbidities, care setting, and follow-up time. The aim is to understand how the data was created. Methods such as matching and weighting can help make treatment groups more comparable based on measured covariates. Propensity scores are often used for this. A propensity score summarizes the probability of receiving a treatment based on observed baseline characteristics and can support matching, stratification, or weighting. Regression adjustment may still be needed if imbalance remains. However, these methods cannot remove every source of bias. They depend on what has been measured, how well it has been measured, and whether there is enough overlap between groups. If treated and untreated patients are very different, matching may exclude many patients. This can improve comparability, but it may also narrow the population being studied. Sensitivity analyses help test how dependent the result is on reasonable assumptions. And for example, if exposure timing is uncertain, researchers may repeat the analysis using different exposure definitions. If the estimate moves substantially, that uncertainty needs to be part of the interpretation. So for an observational study to be useful, the question, data source, and analysis plan need to fit together. Variables should be clearly defined, analytical choices should be justified, and the documentation should be detailed enough for someone else to follow the logic. Observational studies can produce valuable evidence from routine care, but only when their limitations are handled directly rather than hidden in the analysis. To learn how Quanticate can support your next trial, email inquiries@quanticate.com or request a consultation through our website. Thanks for watching, and don't forget to subscribe for more practical insights on clinical trials and modern drug development.